Hello, my name is Manolis Kellis. I'm a professor in CSAIL in the Computer Science and Artificial Intelligence Lab, and I'm the head of the Computational Biology Group here at MIT. The mission of our lab, the mission of the Computational Biology Group at MIT is to understand human biology, to understand the circuitry of human disease so we can reverse it. So what does that mean? That means we're trying to understand systematically what are the genes, the pathways, and the biological processes that underlie every one of the biological functions that are responsible for human physiology, for human cognition, behavior, memory, and obesity, cancer, cardiac arrest, the onset of diabetes, the effect of exercise on the body, the immune system, and so on and so forth. So we have a series of projects that are seeking to, number one, understand what are the building blocks of the human genome. Number two, understand what are the circuits that underlie human disease. And number three, how do we modulate these circuits? How do we use AI and machine learning to understand how to design new therapeutics and new drugs and new molecules that perturb that system and that can kind of bring it back to homeostasis after disease. So how do we reverse the circuitry of human disease? I've always thought, as an engineer, I've always loved the beauty of nature, the simplicity of the evolutionary process, and the extraordinary emergence of complexity that comes from that. So when I started my own lab, I was just fascinated to kind of bring it all together from an engineer perspective, from a computational systems level perspective. How do we develop computational methods to even understand the parts list, find where the protein coding genes are, find where the control elements are that are responsible for turning these genes on and off, finding where these control regions are active across hundreds of tissues. How do they evolve across closely related species? So once we understand these building blocks, we can now start understanding the impact of genetic mutations that are associated with disease. How do they impact disease overall? And how do they impact every cell type in the human body, every gene in the human body, every control region in the human genome? So how do we understand the mechanism through which these thousands of tiny little perturbations that are known to be associated with disease predisposition, how do they act? We've had a lot of industry partnerships through the years. We've worked with biotech, we've worked with pharma to tackle three types of goals. The first one is, can we elucidate the biology of an area that they care deeply about? So, if they care about neurodegeneration or cancer or immune disorders, we have basically formed partnerships that allow us to focus our creative juices on the area of biology that they care the most about. And that has been extraordinarily impactful because our researchers and industry researchers are basically working side by side. It's not just a question of sponsored research agreements. It's a question of putting minds side by side, aligned on the same problem, and sort of having these kinds of interactions, these insights that come only when you put people from academia and industry side by side on the table. The second area of collaboration and partnership is actually translating some of these therapeutics, designing and making some of these molecules. And that's a place where, again, pharma can gain enormously from working with academia. This is a place where the discovery process is abridged, where the technologies are from this extraordinary explosion of AI and genetics and genomics and innovation have basically sped up the speed with which the technology can truly make it to a new discovery. And that's a place where having the strength of pharma, the extraordinary libraries and the capabilities of testing new drugs at high throughput can just, again, be so complementary to what we're doing in our team. And the third one is about delivery. It's about understanding the patients. It's about understanding how we can bring data back from clinical trials. So when you have a drug that, for example, fails in a clinical trial, very often that drug could have been successful if the trial was designed in a slightly different way, or if we understood the patient population a little better, if we understood the biology of what that drug is affecting and what subset of patients will benefit from that particular modulation. And I think that's where the true impact could be in these partnerships. Basically, number one, understanding the biology, number two, designing and synthesizing and making the drugs, and then number three, targeting them to the right population. And that's really my ultimate dream, that all of the research that we are having will impact not just one or two drugs in the market, but hundreds of companies, dozens of indications, and thousands of patients, that we can sort of truly make an impact for the whole world. This vision is not just a dream for the future. This is something that we have already started having some initial successes of, and we have a few examples that are showing the power of this approach. The first one is in obesity, where we went from a genetic association where it was sitting in a gene that turned out to have nothing to do with obesity, to ultimately a circuitry that involved two other genes that are sitting 1.2 million nucleotides away, 1.2 million letters away, several genes over, and that turned out to be master regulators of a totally new process that was never implicated in that locus before, of controlling the metabolism of our own adipose cells, our own fat cells. So our adipocytes, our fat cells, can choose to store calories or burn calories. And what we saw is that that strongest genetic association with obesity can be used to uncover this circuit and to flip the switch between storing calories and burning calories. And that allowed us to now, by a single letter change in the 3.2 billion letters of the human genome, with a single letter change, switch fat storing cells into fat burning cells by re-establishing this process of thermogenesis, or heat generation, whereby the mitochondria, the energy producing machines inside our fat cells, can basically be depolarized, causing a loss of the proton gradient and a loss of energy in the form of heat. And then we were able to go into mice and show that with one therapeutic intervention that targets that gene, we could now turn mice into just fat-burning machines. So these mice could eat anything they wanted, they didn't have to exercise, they were just simply burning calories when they were awake and when they were sleeping. So that holds so much potential for reversing the obesity epidemic in our planet and all of the comorbidities that come with obesity. So that's one example. Another example, just a few weeks ago, we basically showed how we can take Alzheimer's cases and controls, risk and non-risk individuals, and take again the strongest genetic association with Alzheimer's now in the ApoE locus, apolipoprotein E, and show that this locus is in fact leading to a loss of transfer of cholesterol. Why is cholesterol useful? Because cholesterol is how the myelin forms in the neurons that protect the neurons. So we basically show that by restoring cholesterol transport, we could restore myelination of the neurons and ultimately cognition in the mice. Again, providing hope for this extraordinary epidemic of elderly individuals developing Alzheimer's, dementia, and all of the extraordinary complications for themselves and for their families. And the third example of a success story is by studying responders and non-responders to immunotherapy for metastatic melanoma. We showed that the responders and the non-responders were different in hundreds of epigenomic control regions. These are the regions that turn genes on and off. And by understanding the upstream regulator that targets many of these regions, we were able to design a combination therapy that allows us to now couple immunotherapy with a new regulator that will shift individuals between non-responders to responders. And when we try this combination therapy in mice, we again showed that there is zero recurrence in the tumors, that basically the mice were becoming tumor-free. So these are the types of circuits, the types of successes that we want to start translating to the clinic, that we want to continue discovering in those partnerships. So it's not just a dream, it's a reality that we can sort of slowly turn the page on the traditional approach for these discoveries and truly enable dramatic acceleration in the way that therapeutics are discovered. So again, thank you so much for your time. Thank you for listening. Again, this is just a very small glimpse of the extraordinary diversity, both in the field and in my lab, and the extraordinary wealth of projects and students. We're always looking for new students. We're always looking for new folks to join the lab. We're always looking for new partnerships. So if you're listening to this video, please reach out. You can contact me directly. You can contact my students. And I really encourage you to now turn and watch some of the videos from the interviews with some of the students in the group that will take you through different projects well beyond the small glimpse that I gave you here. So thank you so much, and then looking forward to meeting you in person.